All right, guys, so you may have seen this morning that I posted an assignment on Google Classroom called Virtual DNA Lab. You do not need to get this pulled up right now. All you need with you is a sheet of paper and a pencil, as well as some colored pencils. But I do want to run through this assignment that you will need to be completing over spring break. So there are essentially two parts to this assignment. The first thing we're going to do today is draw our DNA strand. That's the first part of this lab that you're going to go ahead and get out of the way. But you can also see that there's a pretty lengthy passage on this assignment, as well as some questions on the back page. After class today, you guys are going to be responsible for finishing up these conclusion questions based on that passage that we read, as well as this structure we're about to draw. So there are, let's see, 14 questions. Some of them are just simple one-word answers. So I promise you, you can get these done. You've got all weekend, all of next week, and all of next weekend to complete these questions. So I just need them by 8:30 on April 5th, that Monday we come back from break, right? But all you need to do today is get out your pencil, a sheet of paper, and something to color with. So I'm gonna get that pulled up on my end. Now, Samuel asked a good question. Samuel said, how good does the drawing have to be because I can't draw? Listen, you guys have seen my drawings before. You know I am not an artist. If I can do this, so can you. It does not have to be perfect as long as I can see that you tried. Okay? So the very first thing we're going to do today is draw a key that we're going to be following as we create our nucleotide structures. So at the very top of one sheet of paper, I just want you guys to write down key. All right, these are the keys we're going to be following as we create our nucleotide. Now, can someone remind me, what are the 3 components of a nucleotide we need to know? So there are 3 parts to a nucleotide. What is that very first structure we need to know? Starts with a P. You can type
Phosphate.
A phosphate, right? So that first part of a nucleotide we're going to be drawing today is a phosphate group. And today, when we draw our phosphate, we're just going to draw it as a simple circle with a P in the middle. Eunice, Dion, do you think we can draw a circle? I think we can. Yeah, okay, we got this. All right, guys, and you're going to color that phosphate in brown. Now, if you do not have a brown colored pencil or crayon, feel free to use some sort of substitute, as long as you tell me on your key that you made that substitution. All right, what is the second part of a nucleotide we need to know? So we've got our phosphate, what comes next? Type it in the chat. Yes, Ariel got it. It's a pentose sugar. But I actually want to take us one step further. So we're working with DNA today. Can someone remind me, what does DNA actually stand for?
Deoxynucleic acid.
Okay, we're close. Deoxy what?
Deoxy.
We're almost there. See if we can type it in the chat. Do either of you know? Deoxy. Yeah, Aaron got it. Deoxyribonucleic acid. Now, you were close, Ian. I'll give it to you.
Yeah, sorry.
So yesterday, we talked about RNA, right? And we said that the sugar that's found in RNA is called ribose. So if the sugar in RNA is called ribose, what do you think the sugar in DNA is called? Yeah, Aaron says deoxyribose. So we're going to say that our next structure is a deoxyribose sugar. right? And as Ariel said, that is a pentose sugar because it actually has 5 carbons in it. So today when we're drawing our sugar, we're going to draw it as a pentagram, a little house shape. So make sure it has 5 edges. We're going to put an S in the center, and we're going to color that sugar purple. All right. So now we've got our phosphate group, we've got our deoxyribose sugar. What is the third part of the nucleotide we need to know? We've talked about our A's and our T's and our C's and our G's. What are those structures called? Bases. Awesome, Becky. And specifically, what type of base? We're going to call it a what? A nitrogenous base. Great job, Erin. So we call it a nitrogenous base because it has a nitrogen group in it. Right? And there are 4 different options of bases we're going to be drawing today. That first base I want you guys to write down is adenine. All right, and we're going to draw adenine today as kind of this front-facing arrow structure with an A in the middle. And we're going to color that adenine in red. Right, now what base do we know that typically connects with adenine?
Thymine.
Thymine, exactly. So if you had to guess, what do you think thymine's shape is going to look like?
Like something that like the puzzle piece for.
Oh.
It will be like the negative version per se. Like the space will be negative.
Yeah,
right?
so we drew like an outward facing arrow for adenine. So our thymine's going to be
An inward facing arrow.
Inward facing arrow, right? So we know that we could connect these 2 pieces, as Ian said, like a puzzle piece. All right, and we're going to color thymine today green. All right, the third base I want us to draw today is guanine. And we're going to draw guanine. Oh, that's ugly. I'm going to try that one more time. Or 2 times. Looking like this. So draw your G in the center of that shape. And then we're going to color it in blue. Right, and what is the name of that last base that connects with guanine? Type it in the chat. What do we call that last base? Cytosine. Thank you, guys. So same thing, we're going to draw our cytosine over here with a shape that is opposite to guanine's. So it's going to look like this. And we're going to color our cytosine yellow. All right, I'm going to give you guys about 20 more seconds to finish writing down that key. What's up? The purple one is, I just drew an S to represent a sugar. Yeah. So that is our pentose or our deoxyribose sugar. Yeah, so this can be anywhere. This is just your key. I would recommend using 2 sheets of paper, one for your key and then one for the actual DNA strand. You can probably fit it on there.
Can I just have the key on like the back of the thing?
Yes, that's totally fine. If you can fit it on the back, that totally works. All right, guys, I've got one more thing for us to draw before we draw our strand, and that is a model nucleotide or an example that we're going to follow when we're making our strand. So this is our model nucleotide or our example. Dion says, my fingers hurt already. Whew, you're in for a treat. Here we go. So guys, whenever we draw our nucleotide, I always think it's easiest to draw the phosphate group first. So whenever you're drawing your nucleotide, you're going to draw your phosphate here in the top left corner. So right now, just draw a simple circle with the P inside. And I'm gonna follow my key and color that phosphate group in brown. Right? The next thing I'm going to do, so we actually have to form some sort of connection points between each of our groups. And to represent those connection points, we're going to draw some bonds as just simple, straight black lines. So anytime you see a black line connecting these two groups, that's going to represent a bond holding those parts together. Right, so our first bond is going to connect our phosphate group to the top left of our deoxyribose sugar. So connect the phosphate to the top left of your deoxyribose sugar. And I'm going to color my sugar in purple. If you want to save your coloring for later, you're more than welcome to do that. So guys, if it's easier for you to just draw down the structure in black and white right now and then color it later, that's totally fine. Yeah. I want it to be pretty. All right, then we're going to draw one more bond connecting the top right of that sugar to the last part of our nucleotide. And what is the last part of the nucleotide I need to draw?
You need to put a nitrogenous base.
I need to write a base, right? Now, I can pick any of these 4 since it's just an example. Does anyone have a favorite base they want me to include? Pick your
Guanine.
That's so
Because
interesting. Someone in my last class chose G too. Wonder why
the reason why I chose G is very simple. It's the first letter of my last name.
Oh, that makes sense. We should have done a C for Collins then. All right, guys, so go ahead and draw down your G or your guanine. And since it is guanine, we're going to color it in blue. All right. And that is one nucleotide. So we're going to follow that model for every single nucleotide we draw today. But we're going to be drawing a lot of nucleotides because we have to form an entire DNA strand. So Dion, I know you said your fingers already hurt. Maybe take about 20 seconds to stretch them out because we're going to be drawing a lot more where that came from. Take about 20 more seconds and finish up your model nucleotide, and then we're going to move on to the next. That's good, Eunice. I'm sure it's perfect. As long as you can understand it, it's your key. All right, guys, and if at any point today I'm moving too fast and you need me to slow down, please just let me know and we can slow down. All right, so it's time. Everyone get ready, stretch those fingers. We're about to draw some DNA. So I would recommend, if you do not have like the majority of a piece of paper, I would either turn to a clean piece or turn your paper over just to make sure you have enough room. I think yours is going to be good though. All right, and at the very top of that piece of paper, I want you guys to write down that the strand we're going to be making today, or at least strand 1, is CTGA. Now remember that DNA has 2 strands, it's double-stranded. So I do want you to leave some space beneath that strand to fill in our complementary strand number 2 later. All right, but now it is time to draw our 4 nucleotides. So again, I always think it's easiest to start with our phosphate. So in the top left corner, I want you to draw just a simple circle with your P in it. So that is your phosphate group. And since this is our phosphate group, we're going to color it in brown. Ooh. All right, and we're going to follow the example that we just made. So off of that phosphate group, I want you to draw just one single bond and connect that bond to your sugar. All right, and you're going to color in that sugar purple. All right, so then we're going to draw one more bond connecting that sugar to our very first base, which is a cytosine. So now following that key, we're going to draw ourselves a cytosine.
Is it fine if it gets a little bit tilted?
You're good. Like I said, I do not expect this to be perfect as long as it's neat and you are trying your best. Right, so take about 10 more seconds and write down that base attached to your strand. Hello. All right, but we still have 3 more nucleotides to draw. So
You're lagging for me. Yeah, you froze, miss.
okay,
Again.
just looking at one side of that ladder. Oh, I froze? Oh,
Yeah,
no.
you froze.
Ah, am I back?
Yeah, you're back. You're back.
Okay. So right now we're working on just one side of this DNA strand. So we need to find somewhere to connect our phosphate group to. Where do we think it's going to go?
Downwards.
Oh, okay. So
Because you can't connect C with T, so like you have to go downwards.
you're right. We can't connect our T over here. That doesn't make sense. So Ian's right. We actually have to go down. We have to connect our phosphate group to the other side of our sugar. I'm actually going to draw that a little bit smaller. And we're going to color it in brown. And now it's time to start that nucleotide all over again. So we're going to do that same thing. We're going to connect our phosphate to our sugar. And then connect our base, except this time we're not drawing a C. We're drawing a thymine. So following our key, that was that inward facing arrow. You're going to write down a T. All right, and I'm going to keep moving on just to make sure we have time to finish this. So again, you're going to draw another bond at the bottom of that sugar. Draw your phosphate group. And then same thing, you start another nucleotide. So draw another sugar. And now this time our base is a G or a guanine. So this is that blue shape we drew up earlier at the top. Ah, I really can't draw this one. All right, and we're going to color those in. So phosphate is brown. Sugar is purple. And then guanine is blue. All right, and we've got one more nucleotide to draw. So again, I'm going to draw one connection point at the bottom of my sugar. Draw a P for my phosphate. Connect my sugar. Ooh, my drawing is getting real sloppy. And then draw my last base, which is an adenine. All right, I'm going to give you guys about 30 more seconds to finish filling in that strand. So your strand 1 should look just like mine on the board right now. I'm sure it actually looks better than mine, if we're being honest. Dion says nope. I'm sure it looks great, Dion.
This looks very scientific. Like,
Oh.
I don't know if you guys can look at it.
Oh, that looks perfect.
But it makes me feel very scientific having this like that.
Very scientific. I think it's great. All right, guys. Now, the next thing we're going to do is we actually have to draw the second strand of our DNA. Now, you might be thinking to yourself, perfect, I'm on autopilot. I've got this. But I'm going to warn you, don't draw that second strand just yet, because we're actually about to see something different. Okay. Now, we know how to connect our bases, right? We know about complementary base sequencing. So using our strand 1, what should the second strand of our DNA look like? So match up those base pairs. Go ahead and send it in the chat. What is the second strand going to look like? All right, I'm seeing the answer from Dion. And I agree, right? It's going to be G. Oh, that's not a letter. Hold on. G, A, C, T. So now we're going to draw that second strand, except this is where things get a little bit different. Now, in math, maybe in geometry, maybe in algebra, have you guys heard the word parallel before? Maybe parallel lines?
I heard it in art.
Okay, you've heard it in art. That works. So what does it mean if something is parallel?
Like
the two lines.
Yeah, the 2 lines never touch. So 2 lines that are both
right.
Yeah.
They're actually running in the same direction, right? But what if I told you that DNA is actually anti-parallel? Whoa. What do you think?
It means like they cross over each other.
They intersect.
Okay.
Or they touch.
If the lines touch or cross over, doesn't that mean it's perpendicular?
No, because it's like it touches and then it leaves,
Okay.
kind of like how the DNA looks. It
Okay.
like crosses, leaves, and it still looks like a mirrored version of the other one.
Oh, a mirrored version of the other. Okay, so if we said that parallel lines are running in the same direction, what do you think anti-parallel lines mean? They're running in what?
The opposite.
In the opposite directions. So our anti-parallel lines are actually going to look like this. So we just drew our first strand running this way, which means our second strand is actually going to be, whoa, running this way? Oh man, this is going to be tricky.
Oh,
Ian says, uh-oh. So, okay, guys, I know this is going to be crazy, but trust me, right now, I want you to actually turn your piece of paper upside down, right? So just flip it upside down. I'm going to flip my iPad upside down. You won't be able to see it on your screen, but I'm now going to be drawing down here in the bottom or in the top left corner or the bottom right. Dion says, do I turn my computer upside down? If you can, that works. All right, so now we're going to fill in the opposite DNA strand, but we're going to do it anti-parallel. So we're starting at the other end of our strand, right? But don't panic just yet because nothing's actually changing. We're still drawing our nucleotide the exact same way, which means the very first piece I'm going to draw is still my phosphate here in this bottom or in this top left corner. So I'm still drawing a phosphate. Right? I'm still connecting it the same way as I did. And I'm going to draw my sugar. Except this time, as I get close, what do you think I'm going to do with my base? So this time the base I'm drawing is a T. What do you think it's going to connect to?
the A.
The other A. So remember, those shapes are actually complementary, which means those shapes can line up perfectly, just like that. Right, so now I'm actually going to just draw the backbone of my structure first, and then I'll go in afterwards in color. Right now, I just want to make sure that we have time to finish this strand. So now I'm going to do the exact same thing. I'm still drawing my next phosphate group. I'm drawing a bond connecting it to my sugar. And then I'm going to connect that sugar to my cytosine. Right, same thing, I'm going to draw another bond connecting my sugar to a phosphate. I'm going to draw another bond connecting that phosphate to my sugar. Then I'm going to draw another base connecting my A to my T. All right, then same thing here at the end. I'm going to draw one more phosphate group. I'm going to draw my last sugar. And then I'm going to connect my base to my C.
Are we supposed to draw the second one too?
Yes, please. Yep, draw both strands. Now, as you guys are drawing this, I'm going to start coloring in my strands according to that key. So again, my phosphates are going to be brown. My sugars are going to be purple. My thymine is green. My adenine is red. Cytosine is yellow. and guanine is blue. And guess what, guys? You just finished your DNA strand. Right? So take a few more minutes, finish that drawing. But what do you notice about the directions of our sugar molecules? And the direction of those bases? They're actually running in the opposite direction, right? Right? Because that is what we describe as anti-parallel. So DNA is anti-parallel, meaning those strands actually run in opposite directions. So earlier this week, we described that DNA structure as kind of like a ladder. And you can kind of see that here, where these sugar and phosphate groups form those outer rings, or outer handles, or no, I should say outer rungs of the ladder. And the bases form those handles that you crawl up. All right, guys, how are we feeling? We just drew a DNA strand. I know we're probably catching up on coloring right now. Oh no. Samuel said if my drawing was bad before, it got exponentially worse when I started drawing upside down. I think you could probably say the same about mine. It didn't look too great. But again, as long as I can see that you tried, I'm sure it's going to be beautiful. It really does not. I'm sure it looks great. Let me see it. Oh, it looks good. Just make sure you draw those connecting points from the shirts. Dion says, yours looks like a masterpiece. Well, remember, I do this multiple times a day for multiple years now, so I've gotten pretty good at drawing my DNAs, okay? But guys, that's all I've got for you today. So again, this weekend over this break, you guys are going to be using that drawing as well as the passage that's on the assignment on Google Classroom to answer your conclusion questions. So there are, let me see, 14 questions waiting for you, but some of those won't take you long at all. Okay, you've got all of this weekend, all of next week, all of next weekend to complete this assignment. I just need it by April 5th. I'll write that down. At 8:30 a.m. All right, so this lab is your only homework over the break. What questions do you have for me before this bell rings? Yeah, go for it. Oh wait, is this, oh yeah, April 5th is the Monday. I almost got confused. Yes, Samuel, great question. So what you guys are gonna do with your drawing, just snap a picture of it on your phone and upload that picture with your lab to Google Classroom. So on Google Classroom, there should be 2 attachments. One, there should be your picture that you just drew, as well as the Google Docs worksheet where you're going to type your conclusion questions. All right, if you guys don't have any other questions for me, you are good to go. Have a wonderful day, and I will see you guys next week. Of course, Jennifer, anytime. Now, Ian and Apostolos and Lauren, if you actually want to stick around, I can go through that homework quickly right now, if that works for you. All right.